Okay, hey everybody. So this is the recording for the lecture that is scheduled for Labor Day, and I figured this would be easier than meeting, obviously, on Labor Day. So I'm just going to jump right in. Okay, so you should be able to see my PowerPoint now. This whole chapter is just about what cells do basically over their lifetime and then how they divide. So we're going to be talking about some general cell cycle stuff and then specifically spending some time on mitosis. I'll try not to go too fast. I tend to do that when I'm just talking to a computer screen. So I'll kind of take my time here, but you can always obviously go back and watch this as many times as you need to. Okay, so what are we talking about when we talk about the cell cycle? It's basically just, as I've said here, a series of events. It involves the growth of a cell and a cell dividing to make a new cell. So cell division and cell growth are the two main processes involved in the cell cycle. During the cell growth process, there's a really important part, DNA replication. So in order to have a new cell, when the cell divides, both of those cells are going to need DNA. So the DNA has to be copied, basically, in order to go into the new cell that's being made. This is generally divided into two overall stages of the cell cycle, interphase, which you see over here, and then mitosis. So mitosis is specifically the cell dividing. Interphase is basically everything else. This is where a cell spends the vast majority of its time in interphase. It's growing, it's replicating DNA, then it's getting ready for mitosis, but mitosis is pretty quick. Okay, so cell growth, DNA replication, and cell division. That's what we're going to be talking about. The purposes of the cell cycle, cells need to do certain things. These are the three main reasons why cells will grow and divide. This is mostly about cell division. So reproduction, single-celled organisms, prokaryotes, like bacteria, they reproduce just by splitting in half. So they make clones of themselves. So reproduction is one purpose of the cell cycle for single-celled organisms. And you can see that picture here, this little purple bacterium just splitting in half, making another one of itself. And then growth and development. So we're not single-celled, we're multi-celled. We can't just divide by splitting in half, but we still need our cells to divide so we can grow bigger. Multicellular organisms all develop from a single cell. So at one point, we were all just one cell, a zygote. And from there, now we're made of about 37 trillion cells. So those cells had to divide many, many times to create the adults that we are today. So growth and development is the second purpose of the cell cycle. And closely related to that is tissue regeneration. We have cells throughout our body at all times that are dying or in the process of dying. So we need to replace those. So dead cells being replaced is the idea behind tissue regeneration. So you've probably heard that you lose skin cells all the time. Obviously, if you kept losing skin cells and you weren't making any new skin cells, that would be a bad thing. So these dead cells have to be replaced as they die off. That is tissue regeneration, and that occurs just by mitosis, by cells dividing, making more of themselves. Okay. The cell cycle. So here we have a nice graphic of what the cell goes through, the different stages of interphase, and then we have the mitotic phase as well. So you can see this is representative of how long a cell stays in each phase. So like I said, interphase is where a cell spends most of its life. It's mostly growing, making more DNA, growing some more, and then it divides. But that's a pretty small piece. The mitotic phase is where there's actual cell division. It's a pretty small piece of their overall lives. So we're going to talk about what happens in each of these stages of interphase and then go into detail on this pretty small slice of the pie here, mitosis. Interphase involves G1, S, and G2. Those are the three phases. So G generally stands for growth. When you think G1, that's kind of the first phase of cell growth. So you can imagine a newly formed cell, it has to grow. That happens in G1, down here, G1. The S phase, really critical phase. This is where DNA is copied, replicated, synthesized, however you want to think of it. So there's DNA in the cell already, obviously. It has to be copied in order to be sent into the new cell that will eventually be formed through mitosis. So S stands for synthesis. That is DNA synthesis, the DNA replication phase. You definitely want to know that the S phase of interphase is where DNA is replicated. And we'll go into a little bit of detail here in a minute about a little bit of the structure of DNA, kind of how that happens. And then the G2 phase is another, it continues to grow. The cell continues to grow, and then there's also a DNA check. So that DNA was synthesized. Hopefully it was done correctly. That's what the DNA check is for in G2. Right before the cell goes into mitosis, the cell will check the DNA to make sure there aren't any errors. If there are some problems with that DNA synthesis, there's a number of things that can happen, and we'll talk about those at the end of this lecture. So G1, S, G2, those are the three things that are happening, the three phases of interphase, and those bullet points under each there are the main things that are occurring in each of those phases. Okay, so we're going to focus, I'm not really going to talk much about G1 or G2. I'll touch on G2 at the end with the DNA check idea. But right now I want to focus on the S phase, which is the big one of interphase where chromosomes are copied or DNA is copied. So I'll talk about some DNA terminology here, chromosome being one of those terms. So we're going to start out with that. If you're not familiar with this DNA terminology, it can be a little bit confusing. As you see, these three words all start with chrome something, chromatin, chromosome, chromatid. Make sure you keep these terms separate or straight. Understand what they are because I'll be using them during this lecture as well as when we get into looking at the specifics farther down the line of how DNA is actually copied, the whole process of DNA replication, which we're not going to get into here. This will just be some basics. We're just going to say, okay, the DNA was copied and move on. Later on, we'll look at how that actually happens. Okay, so first, the first word here, chromatin. This is uncondensed DNA. It's not a term we're typically really familiar with. Let me move my face here. We're used to seeing DNA as chromosomes, right? That's actually condensed DNA. So it's been condensed down, and it gets condensed down in order to easily split it into two separate cells. Otherwise, it's kind of this long, uncondensed form of this really long nucleic acid polymer. So condensing it down into chromosomes is an easy way to separate it into separate cells. So chromatin is uncondensed DNA. A chromosome is condensed DNA. It's just condensed down. So same code, same genetics, same genes. It's just condensed. We'll mostly be looking at chromosomes. But on some of these images, you'll see some uncondensed DNA, which is a good indicator for what stage the cell is actually in. And then chromatid, to make it more confusing, one letter difference from chromatin. A chromatid is 1/2 of what we typically think of as a chromosome. So if we think of a chromosome as these two pieces, a chromatid is one side of that. So one side of that X. When you have two of those together, they're called sister chromatids, and they're exact copies of each other. So what's going to happen during the S phase of interphase? You'll have basically 1/2 of this in a cell. It'll copy itself, replicate itself, and then you'll have a sister chromatid to go along with it. And then we'll talk about what happens to that in mitosis. But these are exact copies of each other, same genes, same alleles. Well, we don't know what alleles are yet, but the exact same information is on both sides of those. So sister chromatids are exact copies. Okay, a few more terms on the next slide. Oops, sorry. Okay. Centromeres are, if we think of this typical shape of a chromosome, the centromere is kind of the waist there, where they're connected, where those sister chromatids are connected. That's the centromere. You can have, you have centromeres when there's just one copy of the DNA as well. So this single chromosome here. The terminology gets a bit confusing because this is considered a chromosome. So this one on the left here, I hope you can see my mouse, my cursor. I think you can. And then this is also considered a chromosome. You can see at the bottom here, one chromosome, one chromosome. The definition of a chromosome, it's defined by the centromere. So if there's one centromere, we call it a chromosome. The difference is this chromosome, there's only one copy of the DNA. For this chromosome, there's two copies of the DNA because you have two chromatids. Also, at that center area at the waist, there's something called a kinetochore. It's a protein structure. The main purpose of that is for the next term to attach to it. The spindle microtubules are these blue lines shown here. Those are going to attach to the kinetochore. And they will pull those two halves of the chromosome apart. So they'll pull the sister chromatids apart during mitosis. So those spindle microtubules have to attach to either side of the chromosome in order to pull them apart. Okay, I think that's most of the terminology for now. That'll at least get us to where we need to be when we're talking about the different stages of mitosis here. This is just sort of a simple image of what happens during chromosome duplication or the S phase of interphase. So like I said, there's a lot that goes on to actually make that copy, but we're not going to go into that just yet. We're just going to imagine that, boom, magically you have two copies of DNA. So you're going to start out in the cell. So before S phase happens, this would be, say, the nucleus of a cell. You have one chromosome here that needs to be copied. So an exact copy needs to be made of that. So chromosome duplication happens. That's what we have here. The chromosome is duplicated, and now you have two copies of the exact same information, genetic information. Those are the sister chromatids. Okay, and one of these is going to go into each. When this cell splits in two, one set of that genetic information is going to go to one cell, and then one set is going to go to the other cell. That's why we need to have two copies of the DNA. Okay, back to this graphic of interphase. So we were talking about the DNA synthesis process here. And this is just obviously a very rough, unprofessional addition to this. But here we have, this is just I think a helpful way to think about how much DNA or how many chromosomes are in a cell at any given point, how many copies of DNA there are, I should say. So in the G1 phase, this is kind of a new cell, it's growing. It only has one copy of DNA. During the DNA synthesis phase, that's where it copies it. So now there's two copies of DNA, of all of the DNA. This is just looking at one chromosome. So then you have two in the G2 phase, you still have those two because the cell hasn't split yet, right? So you still have two copies of DNA in G2. This area is where the cell is going to split. So we're going to go back down to one copy of DNA in each of these cells. So we're starting out with one cell. We're going to end up with two cells. We're starting out with one copy of DNA. So we have to make another copy and then split one of those into each of these new cells. I hope that makes sense. Okay, into mitosis. This is where we're going to spend most of the rest of this lecture. Mitosis and cytokinesis, those two processes together make up the M phase. I usually just refer to mitosis in general. Cytokinesis is the actual splitting of the cell. Mitosis is everything else that kind of has to happen before that. Okay, so we're going to get into the different stages of mitosis. There are 5 stages in all. Again, it's a very small slice of the pie when we're talking about how long a cell actually spends in mitosis. It's not very long compared to the rest of its life cycle. These are the names for each of the phases. On the next few slides, I am going to tell you what happens in each phase and then show you images or diagrams basically of what's happening. So we have prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So it's going from right to left here. It's a little confusing. We start with prophase because it's basically based off of this cell cycle. So we're starting here on the right and going to the left. That's why prophase is on the left here, and we're going all the way to telophase and cytokinesis. Okay, so let's go through each of these stages. I have bullet points for what happens in each, and then I'll show you some diagrams and images. Okay, so this is the order. You want to remember the order: prophase, prometaphase, metaphase, anaphase, telophase. When I learned mitosis back in the day, I didn't learn prometaphase. That's kind of been added in. So a helpful acronym for me, and I just throw prometaphase in now, is PMAT: prophase, metaphase, anaphase, telophase, PMAT. And you just have to stick this one in between the P and the M. Okay. Prophase, we have the chromatin, which remember is the DNA that's in this long uncondensed form. Chromatin is condensing into chromosomes. That's one of the main things that happens during prophase. So it has to condense in order to make it easier to split. So if you're trying to split a bunch of DNA, having it in this complicated tangle of DNA is not going to be easy. So that's why it gets condensed into these chromosomes. And then you have the mitotic spindle forming, which is where these spindle microtubules that I mentioned earlier come back into play. And those are sort of the tracks, like I said here, on which the chromatids will separate. So that mitotic spindle will pull the two sides of that chromosome apart onto either ends of the cell. So you have to have these spindle microtubules attaching correctly to the chromosome in order to split it correctly. Prometaphase, the nuclear envelope is starting to break down. So this is all happening, remember, DNA is in the nucleus. In order for half of the DNA to go to one side of the cell and half to go to the other side, that nuclear membrane, the envelope, needs to break down so the DNA can move. So the nuclear envelope breaking down is a big step or an important step of prometaphase. And then we have, more attachment of the spindle microtubules to the kinetochores. Okay, that's prophase and prometaphase. Let's look at those in diagrams and then on some images up here. So this is interphase. On the left here, we're still at the very end of interphase before we have started the mitotic phase. So you can see in this diagram, it's really helpful to see that the chromatin, which is uncondensed, it will be really hard to split that DNA evenly to either side of the cell. So that's why chromatin gets condensed into chromosomes, which you see here in prophase. So now the DNA is all nicely organized and condensed into our chromosomes. That'll make it a lot easier to split into the eventual 2 new cells. Mitotic spindle is forming. And then prometaphase, you have those spindle microtubules attaching. You can actually see here how they're attaching in this diagram to the chromosomes. And they're going to tear each half of those apart. And the nuclear envelope is breaking down in prometaphase. So you can see that breaking down, whereas here we have the nuclear envelope in prophase still mostly intact. Okay, so prometaphase. And then we'll talk about the last few stages now. So metaphase, these last few stages are pretty simple and straightforward, actually. So metaphase, chromosomes are going to line up along the middle of the cell. When you think meta, think middle, MM. Metaphase, chromosomes are lining up along the middle of the cell. They get all aligned. I'll show you what that looks like in a minute. Anaphase, that's where the two halves of those chromosomes, the sister chromatids, get pulled apart. They're going to separate and move to opposite ends of the cell. So they line up all in line, and then each half of those chromosomes gets pulled apart. So we're at anaphase, and then telophase. You have new nuclei starting to form. So we're going to have two cells eventually. So we have to have two, what we call daughter nuclei forming. And the chromosomes already start to decondense because they've moved to where they need to be. So they'll decondense back into that long stringy form of DNA, chromatin. And then cytokinesis is where the actual splitting happens. In animals, there's something called a cleavage furrow that develops. I'll show you what that looks like, but the cell is basically, if you think of a cell, kind of a circle like this, it starts to pinch in the middle. That's the cleavage furrow, and then it'll split apart. In plants, it's a little different. They have a cell wall, which makes it hard to pinch in like an animal cell could. So they're pretty solid. And instead of a cleavage furrow developing, you get a cell plate. And I'll show you what that looks like. And then that'll actually grow in the middle of the cell and divide the two. Okay, here's diagrams of those last few stages of mitosis. So here, metaphase, we have the chromosomes lining up along the midline. And the spindle are still attached, right? The mitotic spindle, we have those microtubules attached to the chromosomes. That's what's going to pull them apart in anaphase. So here, moving over to anaphase, you can see how those chromosomes that each had two chromatids, each of those chromatids, one moves to one side of the cell, the other moves to the other side. And those spindle microtubules are what is pulling those chromatids. All right, and then this is an animal cell we're looking at. So you have the cleavage furrow developing where, like I said, the center kind of pinches in and eventually separates the two cells. A lot of times you'll see telophase and cytokinesis. Those are kind of grouped together because they all happen, those processes all kind of happen almost simultaneously. So it's hard to separate them in time. So you'll see telophase and cytokinesis linked together a lot. Cytokinesis is technically the actual splitting of the cell, so that development of the cleavage furrow or the cell plate. Let's see if this video works. So I had, hang on, I gotta share this screen with you. So I have let's stop sharing that. Okay. It's a very simple video, but it shows mitosis kind of in action, which is kind of cool. Okay, let's share Chrome. Okay, so now you should be able to see this. I'm not going to expand it, but so here we have, this is mitosis in an animal cell. So we have the cell here. It's kind of hard to see, but I'll kind of pause it along the way. There's no sound with it, but I'll describe what's happening. So there you can kind of see the nuclear membrane here. So we're looking specifically at the nucleus of the cell, and you can see the chromosomes starting to condense here. So you can see them developing, right? They almost look like little worms. So that's the DNA condensing from the chromatin into the chromosomes. Pretty cool. And continues to condense, and now you see them start to move, right? You see those chromosomes kind of starting to move towards the center of the cell. So we're going towards metaphase now. It's taking a little while. Okay, there we go. So now you can really see them lining up kind of in the middle there. And then the splitting will happen really quick once they line up. Okay, so there, good, I caught it. So they lined up, and then right when they're lined up, this is time lapse, so it doesn't happen quite this quickly. But now those sister chromatids are being pulled apart. So we're into anaphase. Anaphase, they move to opposite sides of the cell. So you can see it almost looks like a weird spider being pulled apart. So half of the DNA is over here, half of the DNA is over here. Exact same information, genetic information. So these are exact copies. And then you'll see the cleavage furrow developing, I think. Yeah, there we go. So you can see it start to split. And boom. And then you can see the chromosomes decondensing already back into that chromatin. So you can't see them quite as well anymore. Okay, and that's it. So anyway, just mitosis in action. Let me go back to the PowerPoint slide. You can watch that more if you want to. The link's at the top of this slide if you want to go to, these slides are on Canvas. Okay, so there, we've split, we've made an additional cell. So we had one cell, now we have two cells. Two cells are exact copies of each other. They are no different. They have the same DNA, the same genetics. They're just clones of each other. Yes, so this is just a nice graphic showing how animal cell cytokinesis is different from plant cells. So we saw that cleavage furrow developing, the cell pinches in two. In a plant cell, you have this really rigid cell wall. So it's a solid cell wall that can't really pinch together. So instead, a cell plate, which will eventually become cell wall, starts to develop sort of in the middle here and then grows outward. And that's what will eventually split the two plant cells. Here's images, microscopic images of mitosis in a plant. So I'm going to go through a few just quickly, the characteristics you want to look for in slides like this. On the next exam, I'll likely have some images of cells like this where you can tell what stage of mitosis they're in. So here in prophase, we don't see any chromosomes. The chromatin hasn't condensed yet. And then moving into prometaphase, now you do have really distinctive chromosomes. So it kind of starts in prophase, moving into prometaphase, you have distinct chromosomes. Metaphase, you can see this lining up of the chromosomes along the midline of the cell. Anaphase, they're being pulled apart. So these two halves are splitting apart. And then a cell plate is developing. So this is plant cells. So a cell plate is developing rather than the cell being pinched together at that furrow. Okay, and we'll look at some more of these in class as well and in lab. Okay, so that's mitosis. Feel free to go back and kind of look at that again. Obviously, make sure you know the different things that happen in the different stages and what order they occur in. Okay, so now we're going to talk briefly about checkpoints, cell cycle checkpoints. So the cell cycle is very tightly regulated. If it goes wrong, it can cause a lot of issues, including cancer, which I'll talk about briefly after we go through some of these checkpoints. So there's 3 checkpoints you want to be familiar with. Here, this is organized a little bit differently. It's like the cell cycle is shifted a little bit, but here we have G1. That's that first phase of interphase. So we're going to start in G1. So we're imagining this is a newly developed cell. We just have mitosis. It's a new cell. Okay, great. It's growing, and it gets to this point in the G1 stage called the G1 checkpoint, aptly named. There, basically what's being checked for is it big enough? Does it have enough nutrients to continue on in the cell cycle? DNA damage will be checked for as well, if there's any DNA damage. But for the most part, that G1 checkpoint is to determine, does the cell have enough to go on and eventually split into 2 cells? Is it big enough? If it is, it just continues around the cell cycle into the S phase. If it's not, if it needs a little bit more time to grow, or if there's DNA damage that needs to be fixed, it'll go into this G0 phase. which is a resting state. So it'll rest, build up more reserves, grow a little bit more before it moves on into the S phase. Okay, so let's say it makes it past the G1 point, the S phase, the DNA is synthesized, it's copied. So now we have 2 copies of DNA in the cell. On to G2, the G2 phase. The G2 checkpoint happens right before mitosis. Checks for cell size again. Is the cell big enough to split? Does it have enough nutrients, enough reserves to actually make 2 cells? And it also checks for correct DNA replication. So DNA replication, if there's any issues with it, will be checked here. Similar to over here, if there's anything that went wrong, it can be fixed, or the cell can undergo something called apoptosis, which is death. So if it's just too messed up, the DNA isn't good, the cell will basically just die and not go through mitosis to make more cells that also have messed up DNA. And that's the idea behind the G2 checkpoint. So that's a really important checkpoint. There is one checkpoint in mitosis at metaphase. So during the metaphase checkpoint, what the cell is checking for here is if those spindles, the microtubules, are attached to the chromosomes correctly. So those are what's pulling the 2 halves, the 2 sister chromatids apart. If they aren't attached correctly, enough DNA, more DNA might end up in one of the eventual cells versus the other if they're not pulled apart correctly. So that's the idea behind checking for the spindle attachment. Is everything set up correctly in the cell for it to be able to split the DNA in the correct way? So that is the metaphase checkpoint. So you have G1, G2, and metaphase checkpoints. The G1 and G2 are very similar. Is there something wrong with the DNA, and is the cell big enough? Okay, cancer. We all know about cancer. Some of us may know more about it than others. I know a lot of people are affected by this. I'm not going to go into too much detail about it, but I want to go over sort of what cancer is at the cellular level. And essentially, it's just uncontrolled cell division. So it's where a tissue is experiencing cell division when it shouldn't be. So uncontrolled cell division, often with DNA damage. And then those cells get replicated as well. So it can cause a lot of issues from there. This is usually stopped if it starts in well-functioning tissues. It'll be kind of cut off at the head at these cell cycle checkpoints. So those 2 G checkpoints that I talked about, G1 and G2, they will check for DNA damage. They can repair the damage. So the cell can kind of repair some damage up to a certain point in the DNA and then continue on and hopefully have successful mitosis and healthy cells coming off of that. Or like I said, this is the term I just used on the previous slide, apoptosis, which is cell death. So if the cell is just kind of beyond repair and the DNA has been damaged too much or didn't replicated very, very poorly, and there's a lot of errors, the cell will die. It'll undergo apoptosis in order to, like this graphic says, not produce any more damaged daughter cells. This is actually controlled by a specific gene. So the P53 genes have these letter number combos typically, depending on where they're found on different chromosomes. But the P53 gene they found is a specific tumor suppressor gene. So a tumor is the result of uncontrolled cell growth. So it just cells keep dividing and dividing. This P53 gene, if it's functioning correctly in a person, will keep tumors from developing. Obviously, people with defective P53 genes have more of an issue with uncontrolled cell division and thus tumor development. I'm going to let you guys watch this video on your own, this YouTube link here, just in the interest of time, so you can get through this lecture. But do go watch that. when you have a chance. It's just a little more detail on kind of some of the basics of cancer as far as uncontrolled, a product of uncontrolled cell division. Okay, and then this is the last thing I wanted to talk about just very briefly, a syndrome that actually results from unchecked cell cycles. It's called Hutchinson-Gilford progeria syndrome. It's really rare. It is genetic. There's only one gene. So one gene is responsible out of the many, many genes we have that causes progeria syndrome. And it's basically rapid aging, so premature aging. And it's all due to a cell cycle that's just unchecked. So as we get older, a lot of our aging and the characteristics that come along with it are due to the cell cycle causing cells to divide and produce more cells. And then it kind of, over time, we get less efficient at it, basically. So that's, in a nutshell, part of aging. So this unchecked cell cycle basically creates a whole lot of unchecked cell division. So cells are dividing really quickly, a lot faster than they should, rather than going into that resting state, that G0 that I mentioned earlier, which a lot of cells go into. They just keep dividing over and over. So that actually causes premature aging and causes this progeria syndrome. But it is really rare. It's genetically inherited. It's a mutation in a gene. Okay, and you can see the effects of it. A 1-year-old doesn't show many effects, but then by 12 years, a lot of what we think of as kind of symptoms or characteristics of a more aged person is showing up in just a 12-year-old. So the lifespan, obviously, for individuals with progeria syndrome is unfortunately, not very long. Okay. That wraps up. Okay, great. Yeah, that wraps up the information for cell cycle and mitosis. There's quite a few details in there. I've simplified it down somewhat. Hopefully I haven't simplified it down so much that it doesn't make sense, but make sure you know that information that's on those slides. Watch that video that I didn't link to. So this PowerPoint file that I just showed you is on Canvas, so you should be able to get that link. And let me know if you guys have any questions, and I'll see you whatever the date is, the Monday after Labor Day, the 13th, I think, September 13th. All right, have a good one.